Dear students, today I will discuss on two immunoassays, single radial immunodiffusion and immunoelectrophoresis. I will tell you the principles behind the assays and how can we perform the assays in our laboratory. Now both the assays are dependent on antigen-antibody interactions, and antigen-antibody interaction includes bond formation between an epitope present on an antigen and the antigen binding site present on an antibody. And the nature of the bonds are mainly non-covalent, mainly hydrogen bonds, ionic bonds, van der Waals interactions, hydrophobic interactions take place between an epitope and an antigen binding site present on an antibody. An epitope is a specific site present on an antigen molecule. And antibody is called gamma globulin, which is present in our serum. Each immunoglobulin or antibody is composed of two heavy chains and two light chains, and the amino terminal ends or N-terminal ends of the heavy chain, light chain, it forms the antigen binding sites. So each immunoglobulin molecule has two antigen binding sites, or each arm can bind to one epitope. If the epitope is small, then two epitopes can be bound to two antigen binding sites present on an immunoglobulin molecule. Now, depending on the number and nature of bonds formed between an epitope and antigen binding site present on the immunoglobulin, the affinity of that particular antibody for that antigen can be calculated. We can use this formula for calculating the affinity constant of an antibody molecule for an antigen. And the mode is the Ka value, which is called affinity constant or association constant. The mode is the affinity of that antibody for the specific antigen. Now, suppose if we have soluble antigen with multiple epitopes, the epitopes can be of same type, that is recognized by a particular type of antibody, or epitopes can be of different types, that means different antibodies can recognize them. So if we have soluble antigens and specific antibodies at equivalent concentration, then slowly they will conjugate, they will form lattices, and they will enlarge and eventually form an insoluble complex. It will be precipitated out. This is called precipitation. And the immunoglobulins or antibodies which are able to recognize soluble antigens are called precipitins. Using this precipitation reaction, we can detect the presence of antigen or antibody in a given sample. Now, if the antigen is insoluble and there are antibodies which can bind to the insoluble antigen and precipitate out, then this process is called agglutination, and the antibodies which recognize insoluble or particulate antigens are called agglutinins. This reaction takes place during blood group detection, where antigens present on the red blood cell surface are recognized by the immunoglobulin molecules, and then the immunoglobulins bind to the RBC surface and then clumping occurs. And by seeing the clumping, we can say which blood group I have. Now, if we want to draw a precipitin curve, then we have to take fixed amount of antibody and then increase concentration of antigen very slowly. At the equivalence point, there will be precipitation. If we plot amount of antibody precipitated on the y-axis and amount of antigen added on the x-axis, then we will see three zones. Initially there will be less antigen, but excess antibody, since the concentration of antibody is fixed. In case of excess antibody, the antibody molecules will bind to the antigen individually. They will not form a lattice. So no precipitation will take place. Again, when antigen is excess compared to the antibody, each antibody molecule will bind to the antigen separately. Again, they will not form any lattice. But when antigen and antibody are present at equivalent concentration, then they have to share epitopes present on an antigen. So they will compete for the epitopes and they will form a lattice. When the lattice becomes large, it becomes insoluble, it will be precipitated out. So looking at the precipitin curve for any antigen antibody, we can find out the concentration where precipitation starts, okay, and where precipitation ends. This zone is called the equivalence zone. So if we keep the concentrations of antigen antibody between this zone, we can see precipitation, precipitin curve. An antibody-antigen interaction is so specific, it has led to the development of many immunoassays, and mainly the immunoassays are used to detect the presence of antigen or antibody in a blood serum. Now, immunodiffusion can be of two types: radial immunodiffusion or octaloony immunodiffusion. Radial immunodiffusion was discovered by scientist Mancini, and it is called Mancini method. Radial immunodiffusion is an immunoassay where antibody is trapped in agarose gel matrix and then antigen is added in the middle of it in a well, and then the antigen is allowed to move through the agarose gel. Now, as the antigen is moving through the agarose gel, its concentration will decrease, and when it will get its own equivalent zone, it will form a precipitin line. Looking at the precipitin line, we can confirm that yes, that particular antigen is present. To perform the radial immunoassay, we need antiserum, antigen and agarose. We have to prepare 1.2 to 1.5% agarose. Okay, weigh that amount of agarose, take in a beaker in water and then melt it. Then you add the antibody when the molten agarose has a temperature around 45 degrees centigrade. It mix it properly to make the antibody concentration uniform, and then pour the molten agarose containing antibody on a glass slide. Let the agarose to solidify. After solidification, we will make wells on the gel matrix, and then we will add antigen inside the well, and then we will keep it in a moist chamber overnight. So if we don't keep it inside a moist chamber, it will dry up. Now we have to observe the results. Now we can see visible precipitin rings if the sample had and specific antigen for the antibody which is present in the agarose. We can see visible precipitin rings around the wells, and the diameter of the rings increases with antigen concentration.
Ma'am, why single radial immunodiffusion so called?
Yes, it is called single radial immunodiffusion because the antigen or the sample which we loaded inside the well will diffuse radially inside the agarose gel matrix, and between the antigen and antibody only one component is taking part in the diffusion. The antibody is trapped in the gel matrix, so only the antigen is moving. That is why it is called single, and as it is moving radially, radial immunodiffusion. Other methods are there where both the antigen and antibody diffuse together, and that method is called double diffusion method. As we are adding antigen in the well and antigens are moving radially from the well, the concentration of antigen will be higher in this area, in this zone initially. So at higher antigen concentration, no precipitation will take place. Now as the antigens are moving, their concentration decreases, and at this zone, after decreasing, the antigen concentration has reached to its equivalence point. Okay, that is why the precipitin line is observed at this region, and the diameter of the precipitin line can be used to quantify the antigen concentration. So we can use radial immunodiffusion qualitatively to detect the presence of a particular antigen or antibody. At the same time, we can use radial immunodiffusion assay for quantitating the amount of antigen present in a given sample. For that, we have to take known concentrations of a given sample. Okay, you load them in different wells and you load your sample, and then after doing the experiment, you measure the diameter of the precipitin rings formed around each well. Then put the diameter square value on the y-axis versus antigen concentration on the x-axis. You will get a standard curve. Now you take the diameter square of the unknown sample and extrapolating that value, we can find out the antigen concentration of the unknown sample. So radial immunodiffusion can be used quantitatively also to find out the actual concentration of any antigen present in a given sample. And this assay is very sensitive. It can detect the presence of antigen as low as 1 to 3 microgram per ml. Now before performing the experiment, we have to remember a few points, like the molten agarose temperature should be maintained around 45 degrees centigrade. It should not be more than 45 degrees centigrade or below 45 degrees centigrade. Before addition of the antibody, can you tell me why the temperature should be checked before adding antibody?
Ma'am, I think it is because the antibodies are proteins and high temperature might denature them, so they will lose their conformation and we would not get proper precipitin lines with the antigens.
Very good. And it should not be less than 40 degrees centigrade. Otherwise, the agarose will be solidified before addition of the antibody. That is why the temperature of molten agarose is very crucial. And digging the well should be made very perfectly. Otherwise, when the antigen will diffuse radially, it won't be perfect. And we should measure the diameter of the ring carefully. Otherwise, they may give false results. It was discovered by Pierre Graber, and this immunoassay is composed of two techniques, electrophoresis and immunodiffusion. And this is a very powerful technique to characterize antibodies present in a serum. In immunoelectrophoresis assay, first the antigens are electrophoresed to separate different antigens depending on their molecular weight. Then the separated antigens are detected by adding antibody in traps. The antibody will diffuse out and they will form bonds or precipitin lines with the separated proteins. To do the immunoelectrophoresis assay, we need antibody, antigen and agarose. Do you know what is agarose?
Ma'am, I think it is a polysaccharide made up of repeating units of galactose and 3,6-anhydrogalactose, which forms a mesh-like network and acts as a sieve for the separation of the molecules according to their molecular weight.
Yes, and it is able to separate proteins, DNA, since they have pores inside the gel matrix. Now, for doing immunoelectrophoresis, we need to prepare 1.5% agarose gel. So, you weigh the exact amount of agarose, you take it in water and then melt it. Then you pour the agarose solution on a glass slide, allow it to solidify, and then you punch a well on either side of the glass slide. Then the glass slide is placed horizontally in the electrophoresis chamber, and then we will add the antigen or the sample, okay, or blood serum which we want to separate. Now the well should be on the cathode side, because normally the proteins are made negative by using SDS, so they will move towards the positive end or the anode end. So the well where we will load the antigen should be on the cathode side. Now, and normally 10 microliter of antigen can be loaded in a well. Now, PBS buffer, phosphate buffer saline is given for electrophoresis. We have to set the voltage around 50 to 100 volt, and the electrophoresis will continue. And the sample will have a dye, indicator dye. When the dye will travel 3 to 4 centimeters from the well, we will stop the electrophoresis. Now, once the electrophoresis is over, we will take out the glass slide containing gel, keep it there for 15 minutes. Then we have to remove gels from both the traps. Okay, we have to make traps both side of the well. Now we need to add the specific anti-serum in the traps. Okay, suppose the sample you have loaded carrying two antigens A and B. Okay, we don't know their presence, but we want to find out whether a given sample or blood serum is carrying A antigen and A and B antigens. Then what we have to do? We have to add anti-A antiserum in one trough and anti-B antiserum on the other trough. And if the antigens are present in the serum after their separation, they will diffuse. Okay. At the same time, the antibodies will diffuse from the troughs and they will form precipitin lines. One precipitin line will be observed for A antigen and anti-A antiserum. The other precipitin line is for B antigen and anti-B antiserum. This is the gel matrix after immunoelectrophoresis. You can see here the precipitin lines. They are like opaque arc. Since the antigens present in a given sample are separated and diffuse towards the antibody containing trough, they will form a precipitin like opaque arcs. Okay, and counting the number of arcs, we can find out whether an antiserum is homogeneous or heterogeneous. One precipitin line between the antigen well and antibody trough indicates the presence of one antibody in the antiserum. That means the antibody is monoclonal or homogeneous in nature, whereas presence of multiple precipitin lines indicates that the antibody is heterogeneous or polyclonal. And if there is no precipitin line in between the antigen well and the antibody containing trough, then we can say that that antiserum does not contain that any specific antigen present in the serum or sample. Before doing this experiment also, we have to remember a few things, that electrophoresis should be done under low voltage.
Ma'am, why is immunoelectrophoresis conducted under low voltage?
Because the agarose gel we are making here is very thin. If we increase the voltage beyond 100 volt, then it may melt down. That is why we have to do the electrophoresis at a very low voltage. And wells and troughs should be cut properly. Now, immunoelectrophoresis technique has been modified further, and now different variants of immunoelectrophoresis are available, like crossed immunoelectrophoresis, rocket immunoelectrophoresis, fused rocket immunoelectrophoresis, affinity immunoelectrophoresis, etc. Now, in crossed immunoelectrophoresis, what is done? It is a two-dimensional electrophoresis. That means electrophoresis is done twice. Okay. First, immunoelectrophoresis is done to separate the proteins present in a given sample. Then the second immunoelectrophoresis is done in agarose matrix containing antibody. So, immunoprecipitation takes place during the second dimensional electrophoresis, and each precipitate represent one antigen. And from the surface area of that peak, we can find out the amount of antigen present in that given sample. Rocket immunoelectrophoresis is another variant of immunoelectrophoresis which can be done quantitatively, which is used to detect the presence of antigen as well as its amount in a given sample. It is a quantitative immunoelectrophoresis. Here also the antibody is trapped in agarose gel matrix, then antigens are allowed to run through the agarose gel matrix, and depending on their concentration they will form rocket like structure. Measuring the height of the rockets, we can find out the concentration of the unknown sample. We can load known concentrations of antigens. After electrophoresis, they will form rocket like structure. Measuring the heights of the rockets, we can draw a standard curve where we will put the heights of the rockets on the y-axis and antigen concentration on the x-axis. And then from the standard curve, we can find out the concentration of the unknown antigen. Diffused rocket immunoelectrophoresis is a modification of rocket immunoelectrophoresis, and it is also used for detailed measurement of proteins after separating any sample. One more modified immunoelectrophoresis is there. It is called affinity immunoelectrophoresis. Here the proteins are electrophoresed through interaction or complex formation with other molecules which have affinity for the antigens. We have discussed today principles and procedures of two immunoassays, single radial immunodiffusion and immunoelectrophoresis. Single radial immunodiffusion involves diffusion of one component, that is antigen here, and immunoelectrophoresis involves first separation of the antigens, then diffusion of both the antigen and the antibody. And in both the immunoassays, precipitin lines are formed, and precipitin lines are formed when antigen and antibody are present at equivalent concentration. And looking at the precipitin line, we can detect the presence of a particular antigen or antibody in a given sample, and we can use single radial immunodiffusion to quantify the amount of a particular antigen present in a given sample by comparing the result with known concentrations of known protein.